With those, they could closely study and test treatments on other diseases, understand gene functions, create artificial tissues, and more. In modern labs, cell lines are a very important tool because they provide an unparalleled way for us to test things like medicines on real complex biological systems. And having multiple kinds of cell lines derived from multiple kinds of tissues means researchers can choose the one that's most relevant to what they're interested in. Cell lines may or may not be models of a specific disease, but they're derived from a specific tissue, so they still work like a tissue, at least enough to help understand how that tissue works. So if you're studying insulin secretion for diabetes research, you probably want pancreatic cells, for example. Using cell lines also means that you don't have to start your research in humans or animals right off the bat. Since the cells are alive and doing cell stuff, you can test how they'll respond to your experiments really quickly and easily. That might, for example, get you as far as a drug candidate that you can then test in animals and, if all goes well, on humans later on. And it's not just drug development. Every detailed little how does molecule A work with molecule B experiment that molecular biologists do probably needs cell lines at some point. So they're a huge part of how we understand everything in biology that is smaller than a bacterium. Cell lines are even cost-effective compared to other techniques, which is always a plus when you're trying to make your research budget go as far as possible. Researchers can make their own cell lines, or they can buy them from other private companies or nonprofit cell banks.